Good afternoon. Thank you for joining today's webinar hosted by the Alliance for Safe Biologic Medicines. Today we're talking about pharmacovigilance in biologic medicines. We'll be starting shortly. In the meantime, if you have any questions, we ask that you submit them in writing. You should see a text box on the right-hand corner of your screen. You can also email media@safebiologics.org and we'll respond promptly. Good afternoon. I'd like to introduce Dr. Dolinar. He's a practicing endocrinologist based in Arizona, and he's also chairman of the Alliance for Safe Biologic Medicines. We're pleased he can be here today to present on this topic. Thank you, Dr. Dolinar, for your time. I'm going to hand it over to you now.
Thank you very much, and good afternoon, everyone. I'm pleased to welcome you today to our third in a series of webinars regarding biologics and biosimilar drugs. So thank you for joining us today. As you can see on our next slide, the Alliance for Safe Biologic Medicines has been together for over a year, and our membership includes patient groups, physicians, researchers, and industry. We came together with one goal in mind: to improve the lives of the patients we all serve by helping to ensure that their safety is the primary focus of efforts to bring biologics and biosimilars to the U.S. It was in March of 2010 that the healthcare law was signed, and in it was the Biologics Price Competition and Innovation Act. And this tasked the FDA to establish an abbreviated pathway that biosimilars could use to come to the marketplace. To date, we have worked hard to be a source of information for all things related to biosimilars. We think that this is especially important now that the FDA has released its draft guidance last week on February 9th. Everything we do is for the patient who will benefit from these biosimilars. And we're determined to seek solutions that ensure affordability and access, but never compromise on the patient's safety. If you've not done so already, I would strongly encourage you to go to our website, safebiologics.org, and you can see it there on the slide, and it's replete with information that I think will help many in the audience understand biologics and biosimilar drugs. So please check that out if you haven't, safebiologics.org. This is an overview of what we're going to cover today. As I mentioned, the draft guidance was provided by the FDA last week on February 9th. And we'll get into that, and also pharmacovigilance, which is very important, and track and trace of medications, and concrete steps that we can take to improve patient safety. And then at the end, we'll get into interchangeability and drug substitution. As most of you know, the FDA released the draft guidance for biosimilars, and that was last week on February 9th. And I must say, we are pleased, very pleased in fact, but not surprised that the FDA laid out a very thoughtful approach. It avoided a one-size-fits-all solution. It recognized that every application is unique. And it's clear from reading it and going through it that the FDA is determined to remain the gold standard, and that it is also willing to build on the work of others done in the European Union and also Canada who have been working on this issue. The draft guidance suggested that a biosimilar approval is expected to take about 10 months and longer if a determination of interchangeability is requested. And it's clear that the FDA recognizes the challenges around interchangeability. Here's the definition by the World Health Organization. It defines pharmacovigilance as follows: the science and activities relating to the detection, assessment, understanding, and prevention of adverse effects or any other drug-related problem. In layman's terms, quite simply, what pharmacovigilance means is that we want to make sure that if there's an adverse outcome or a bad effect from a drug, from somebody taking a drug, with pharmacovigilance, we want to know that it happened, figure out why it happened, and then ensure and make sure that it never happens again in order to keep our medication supply safe. So in a nutshell, monitoring, determining when problems occur, how to correct them, and making the drug line safe. Tracking and tracing is very important. You might remember last summer there was an issue with the melons, and there's a listeria breakout. Because of track and trace systems in place already, the authorities were able to go back and determine exactly where these tainted cantaloupes were coming from. In fact, the specific farm that they came from. And by doing so, stop further distribution and save lives in the process. Likewise, with medications, it's very important, if there's an adverse event, it's important that it be reported and that track and trace be used to identify it and work on the problem. And as I said, keep our supply of medications safe. We want to detect them early. We don't want patients exposed to unnecessary dangers. And we want a system that's highly responsive. Why is track and trace important for biologic medicines? Well, as you recall from our previous webinars, there's a significant difference between the biologic medicines and the small molecular medicines. As you might recall, our small molecular drugs, they're made via chemistry, via chemical reactions. And over on the left, we've got an example, and that's aspirin, a commonly known chemical drug that we use. And off to the right, you can see some biologics for comparison. The aspirin is only 180 daltons in size, and it's got 21 atoms. But look at, once we move into biologics, insulin, a relatively small biologic, has 51 amino acids, weighs 5,800 daltons, has 788 atoms, much larger than the aspirin. And as you progress through to the right, through somatotropin and onto the IgG1 antibody, you can see how the sizes ramp up significantly. Somatotropin, 191 amino acids, over 3,000 atoms, weighs approximately 22,000 daltons. And you get over to IgG1, over 1,000 amino acids, weighs about 150,000 daltons. We've got over 20,000 atoms. And this is very important. The biologics are very different than the chemical drugs. The biologic drugs are basically grown. They're derived from living cells. And it's very different than the chemical processes that we go through to develop the chemical drugs. The biologics are made by genetically engineering living cells and producing the product, and very different. When you're working with large cells, one of the issues, or large molecules, one of the issues that occurs is immunogenicity. The body identifies it or a part of that molecule as something foreign and then turns on the immune system to attack it. And immunogenicity is a measure of the immune response to a therapeutic drug. And these larger molecules that are being made via the biological process are much more apt to elicit such a response because of their size, because of their complexity. And fortunately, most health authorities do recognize the sensitivity around biologics and biosimilars in causing such safety concerns. I like this analogy. It's a ball of yarn, and in many ways, it's a representation of these large biologic molecules. Because they're so big, minor differences, very minor differences, and we'll show you in future slides, are magnified, and the result can be an important and unexpected response on the part of the body to them. And as I mentioned, biologics are grown. These are not chemical reactions. You know, with a chemical reaction, the example I use, we put together 2 hydrogen with 1 oxygen, and we get water. We always get water. It's a chemical reaction. But with biologics, it's more like farming, where we're actually, in a sense, going through molecular farming to bring these proteins out and bring them to the marketplace. And because no 2 living cell lines are identical, biosimilars will be different than the innovator product, so we will have slight differences there. And because they're different cell lines, they have different starter cells, et cetera. The other thing that I want to point out, when you go from a facility that's making your product and now you want to upgrade it and produce more, mere changing location, changing the process can produce a different quality of protein. And I'll give you an example later on. So these are very finicky, very sensitive molecules we're working here with. And rarely can we see the full spectrum of potential clinical outcomes in clinical testing alone. And thus, we've got to be very careful with these drugs to ensure that patients aren't put at any additional risk. Those of you who are interested in wine, you can appreciate this slide. As I said, the biologics are extremely sensitive to their environment. And like with wine, where the vintage matters, the rain, the soil conditions, the weather, and how they affected that plant, similarly with these biologic molecules and the biosimilars, the environment they're in has an impact on them. So things like transportation, refrigeration, equipment changes, containers, they all can affect the quality of the biologics. These are very sensitive molecules. And adverse events can occur. Let me give you a couple examples here. The first one involves product drift and the medication myozyme. And quite simply, there's a genetic disorder in which the body does not metabolize glycogen effectively. And these people are missing the enzyme that does that. And they can get into serious problems, ultimately winding up with death. They get into problems with low blood sugar reactions. They get into problems with the liver increasing markedly in size. They get into problems with their muscles, and ultimately they die. What researchers were able to do, and I think this attests to the sophistication of the science that we've got available today, the researchers were able to take Chinese hamster ovary cells and derive from them a replacement enzyme to be given to these people so that they could adequately metabolize the glycogen and save their lives, literally. But a problem occurred after developing the drug, and now they went to ramp it up so that they could make a larger quantity, and they changed facilities and set up their new system. And even though they were using the same cell line, they ran into some problems. And the problems occurred when the amount of carbohydrate on the molecule changed for some reason. And that is needed to help this drug get absorbed into the muscles so that it can work on the glycogen there. So the bottom line, this company with the same cell line, with the same researchers and developers, ran into significant problems initially when they went to ramp up on the drug. Subsequently, they have resolved that issue and things are working fine. But the point I want to make is how truly sensitive these drugs are. Also, another example occurred in Europe with Eprex. Eprex is recombinant human erythropoietin. Our body uses erythropoietin to produce red blood cells. This is one of the signals that goes out. Your body turns over red blood cells about every 120 days. Every 120 days, a red blood cell dies, another one gets born. So your blood cells last 120 days, but the body's got to be able to replace them, and it's the erythropoietin that does that. Well, what happened was the company tried to make the same drug that was being made in the United States, the erythropoietin in the United States. And these two companies worked together to bring this about. But unfortunately, problems developed. It's still unclear exactly all the factors that were involved. But the bottom line is this. Besides being very sensitive molecules, sensitive to the environment, they can also cause significant problems. And in the case of Eprex, here's what happened. individuals taking it, some of the individuals taking it developed antibodies to the EPREX, so it blocked the EPREX from stimulating the body to make more red blood cells. Those antibodies also blocked the patient's own erythropoietin. So the patient's own erythropoietin could not function, could not stimulate the body to make red blood cells. And they developed what we call pure red cell aplasia. And with pure red cell aplasia, you've got the ability to produce no red blood cells. Some of these people became transfusion dependent for life. Others actually died. So you can see that some of the very serious consequences that can occur, potentially occur, when problems run awry or things go awry with these biologic drugs. I think it's important to reiterate that biosimilars are not identical to the innovator drug they attempt to replicate. Once again, we're coming out of two different cell lines. They're made by genetically engineering living cells, and because they're coming out of two different living cell lines, they're not identical, and the biosimilars will be different than the innovator product. And that's why they're not the same as generics. Remember, generics, with the small molecular chemical drugs, generics are identical to the brand drug that they go to replicate. With the biologics, they're similar. They're not identical. So we must be able to trace any adverse event quickly, identify its source, identify the product, and determine what the etiology of the problem was. So this means using unique and distinct names and being able to track these appropriately. It also means proper labeling. On the right here, you can see a label that I'm sure all of us have seen when we go to the pharmacy. And it's got all that information on it to help with tracking, accurate tracking of the product. And it's tied into a comprehensive system to report and attribute adverse events. So it's a very important part of the your prescription when you pick it up, the label. I mentioned distinct names as an important component of a track and trace system. As a practicing physician, I need to know with accuracy and specificity the name of a product in order to safely prescribe for my patients. In order to cut down on any confusion, in order to be sure that my patient gets the exact product that we wanted them to receive. The INN, the International Non-Proprietary Name, this has been set up worldwide so that when physicians are communicating with each other, we're using the proper terminology and we're able to speak with each other appropriately. Names are very important, and communication and scientific discourse, I think, is critically important. And we know now that with the FDA guidance coming out and other developments, we know that the global biosimilars market is expected to grow substantially. These drugs will be used around the world, and as we expect, various physicians in various countries are using these medications, and we want to be able to communicate with them. Thus far, the World Health Organization and Japan have laid out clear guidelines that specifically address the naming of the biosimilars. The World Health Organization recommends a unique brand name, and also a lot number should be provided so that the drugs can be traced adequately. And then with Japan, they in their system that they've already set up, they're recommending that the non-proprietary names should be followed as follows. So first follow-on biologic would be follow-on number 1, follow-on number 2, follow-on number 3. And in this way, they'll be able to adequately identify each drug. Thus far in the United States has yet to come out with an outline or formal guidance in regard to naming of these biosimilar drugs. We have additional challenges that can occur. With so few drugs initially out there, and now more and more of them coming to the marketplace, and with the FDA guidance out, this expansion of the drugs will create a heightened sense of urgency to monitor the global supply chain and the manufacturing process. With the biosimilars, we really don't know a lot about the effect at this time of switching a patient from a biologic to a biosimilar drug, especially how a patient's immune system may be affected. We're pleased to see that the FDA seems to be taking a cautious approach in deciding how a biosimilar can be approved as interchangeable. In November, the AMA amended its position on substitution by stating that biosimilars deemed interchangeable by the FDA would be treated as Class A generics and could be automatically switched at the pharmacy counter. I have a tremendous amount of respect for the AMA, and in fact, I'm a member of the AMA. But as a practicing physician, I'm concerned when a doctor is left out of the treatment decision. And I believe that at a minimum, a physician should be notified any time that the treatment he or she prescribes has been changed. I think it's imperative that the doctor be notified. This way, if there's any difference in response after that, I can then look back and say, well, your problem started on July 1st. You had switched medications that day or the previous day. It's important that I know what medications my patient is on. It's important that when I write for a medication, that my patient gets exactly what I write for. If they're going to be switched, I think it's imperative that myself and the other physicians be notified of such. The FDA draft guidance has left this issue open at this time. So the effective implementation of the biosimilars pathway is critically dependent on a strong track and trace system. Without such a system, public health can be at risk. And prior to biosimilars market entry, key policy questions must be addressed with a science-based, transparent approach that seeks the input of major stakeholders and puts patients first. So, very excited about the medications that are now coming to the marketplace. It helps myself and other physicians help our patients in ways that we could not before. The biologics have been extremely helpful, and I believe the biosimilars will play a great role in helping us treat our patients. So I'd like to thank everyone for listening today. I hope it's clear that we must have a strong track and trace system in place as we move to create the biosimilars pathway. The Alliance for Safe Biologic Medicines stands ready to assist the FDA as it moves forward to provide a blueprint to bring biosimilars to the U.S. It's clear from the draft guidance that they will take a thoughtful approach, realizing that one size does not fit all, and placing the emphasis on keeping patients safe. I want to let you know that we will be hosting a forum on February 27th, and we welcome all who took the time to participate today to attend. Information for this event is available on our website, safebiologics.org, and I do hope you have the opportunity to attend. And what I'd like to do now is open up the floor for questions and be happy to take any questions from you. Once again, thank you for participating today and listening to our presentation.
Hi there, Dr. Dolinar. We have a few questions. The first one is, as a practicing physician, how have you seen biologics impact medicines? What do you expect to see in the future?
Oh, I'm an endocrinologist. Insulin, as I pointed out, is one of the many biologics available out there. And it has been absolutely incredible the impact that we've been able to have on patients with insulin and with biologics like that. For example, I'll give you one example. Prior to 1922, prior to the first discovery of insulin, if a patient developed type 1 diabetes, in the past that was known as juvenile diabetes, if they developed type 1 diabetes, 100% of them were dead within 6 months to 2 years, 100%. The diagnosis was worse than being diagnosed with cancer. Insulin's a life-saving drug, and over the years, our researchers have adjusted the molecule, made it more pure, made it now such that it gets absorbed faster, works quicker, works better. So our analog insulins, our biologic insulins that are now out, have made it much easier for the patient to maintain control of their diabetes. And that's just one example, and that's something I work with every day. Thank you.
Hi, Dr. Delenar. The next question, slide 5 mentions the guidance being silent on naming. What are the issues?
Well, as I pointed out, one needs to be able to clearly communicate. And with the, you don't want confusion. The doctor needs to communicate clearly to the pharmacy what drug he wants or needs. The pharmacy needs to provide that to the patient and clearly mark what drug it is. And as I pointed out, the World Health Organization and Japan have already ventured down that road and established processes whereby names are being attached to these various drugs. And at this time, the FDA guidance is silent on naming. I can't speak on behalf of the FDA. I cannot tell you why they're silent at this point. I don't have an answer for that.
Hi, Dr. Delenar. The next question is, how concerned should patients be about substitution of one biologic for another?
Well, this is a new road we're heading down. As I mentioned, let me back up a second. Are you talking about one biologic for another, or biosimilar with another biologic?
Sorry, I think what is meant is biosimilar.
Yeah, okay. As I was going to say, this is a new road we're going down. Because these drugs are not identical, there's the potential risk of immunogenicity. And I think what the biosimilars can bring to the marketplace is bringing a product at a cheaper price and improving accessibility. And I think that is excellent. On the other side, we need to be cautious because this is not the same exact molecule as the original branded biologic. There's the potential for injury or damage occurring. And I think one example, as I pointed out, with that situation in Europe with the EPREX, here you actually had two companies working together to bring that to the marketplace. And because of some problems that developed, some of those patients suffered severely. Some of them actually died. So the potential problems can go from minor all the way up to death. And I think that's why it's so important to have a system in place so that we can identify what drugs are being used, when they're being used. If a problem occurs, we're able to track back and trace which drug it was, identify where the problem was, and then hopefully correct it and keep our medical supply system safe. So the potential out there is significant. Some of these events may occur only rarely. So in other words, when the initial studies are done, when testing a drug, there's a defined population that's used. Now, once they're out into the marketplace and many patients are using them, those rare events can now start to pop up. So the bottom line, we need to stay vigilant, and as long as we do that, I think we can proceed safely. Thank you.
Hi, Dr. Dellner. So we have the last question here. If you could recommend anything to the FDA about their biologics guidance, what would it be?
Oh, the first thing I recommend, please be aware that we're here as a ready source of information and willing to help. We would love to share our expertise and knowledge and work with the FDA as it proceeds down this path. We have brought together a lot of information on our website. We're available to take questions and to help give answers as the FDA and all of us proceed down this path. I think by working together, by having the physicians, the researchers, the regulators all work together, in the end, it's going to be the patient who benefits. Are there any other questions?
You know, hold on one second. We're just getting in one more question.
Oh, okay.
Yeah, here's actually another one. Dr. Dellner, you mentioned that biologics can change over time. What do you think is the proper way to test for this? That's the question.
Oh, well, that's a good question. The testing for that, there are many ways to test drugs, and you know we test them, for example, in the lab. We test them in animals, and then ultimately clinically. I think that the ultimate gold test is the clinical testing. We've gone through the early testing. We've shown it to be safe in animals. We've shown that the structures. We've done all the analytic work. The last part, I think the gold test is the clinical testing. So how does this drug work? How do the patients respond to it? And that's critically important. And the reason it is, at this time, we don't have the ability to adequately predict immunogenicity, immunologic reactions occurring. So the safety challenges are significant. We can't just look at the structure. We can't just look at the chemical attributes. We can't use we can use all these things, but even using all of these, we are unable to adequately predict how's the body going to respond immunologically. So the bottom line, I think the ultimate, the final, the gold test is the clinical testing. And I think clinical testing is critically important. Like I said, because of our limited knowledge, our scientific knowledge regarding predictability. The other thing that makes it so important, when these occurrences occur, they can be quite rare. So you're trying to pick out a needle in a haystack, but yet the consequences can be quite severe. So the bottom line, I think clinical testing, and then after it's out on the marketplace, monitoring, tracking and tracing, monitoring how are the patients responding, how are they doing with this drug now that a large number of people are taking it. Thank you.
Okay, this is actually the final question now. So Dr. Sherman at the FDA previously expressed concern that the existing track and trace system is not sufficient for biosimilars, and do you share similar concerns?
I'm sorry, could you repeat that question, please?
Sure. Dr. Sherman at the FDA previously expressed concerns that the existing track and trace system is not sufficient for biosimilars. Do you have similar concerns?
Yes, I do. I mean, just using that one example being the naming, we've got to set up a system where we can identify these drugs clearly, and then set up a system where we can monitor this. And currently, there's no track and trace system in place to account for the biosimilars. So I share his concern. But this is something that we should be able to fix. We should be able to set it up. We should be able to go forward safely once such a system is set up.
Well, thank you so much, Dr. Noller, and thank you for everyone for tuning in today. As a reminder, we have a lunch roundtable scheduled on Capitol Hill on February 27th. It's at noon. Several members of the Alliance will be speaking and joining in on this event. If you have any questions, please email us or visit our website, www.safebiologics.org. Thank you, and have a great day.